हेलो फ्रेंड्स, आज NCERT क्लास 11th चैप्टर 10 सेल साइकिल एंड सेल डिवीजन चैप्टर रीड करेंगे। सेल साइकिल एंड सेल डिवीजन। आर यू अवेयर दैट ऑल organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristic of cells. Indeed, of all living organisms, all cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cell cycles of growth and division allow a single cell to form a structure consisting of millions of cells. Cell cycle. Cell division is a very important process in all living organisms. During the division of cell, DNA replication and cell growth also take place. All these processes, for example, cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genomes. The sequence of events by which a cell duplicates its genome, synthesis the other constituents of the cell, and eventually divides into two daughter cells is termed cell cycle. All throughout cell growth in terms of cytoplasm increase is a continuous process. DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosome DNA are then distributed to daughter nuclei by a complex series of events during cell division. These events are themselves under genetic control. Phase of cell cycle. A typical eukaryotic cell cycle is illustrated by human cells in culture. These cells divide once in approximately every 24 hours. Figure 10.1. However, the duration of cell cycle can vary from organism to organism and also from cell type to cell type. Each for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases: interphase, M phase, mitosis phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phase. It is significant to note that in the 24 hours average duration of cell cycle of human cells, cell division proper lasts for only about an hour. The interphase lasts more than 95% of the duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of a daughter chromosome, karyokinesis, and usually ends with division of cytoplasm, cytokinesis. The interphase, thought called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication and in orderly manner. The interphase is divided into three further phases. G1 phase gap 1, S phase synthesis, G2 phase gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grow but does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time, the amount of DNA per cell doubles. If the initial amount of DNA is denoted as 2C, then it's increased to 4C. However, there is no increase in the chromosome number if the cell had diploid or 2n number of chromosome get G1. Even after S phase, the number of chromosomes remaining the same 2n. In animal cells during the S phase, DNA replication being in the nucleus and the centriole duplicates in the cytoplasm. During the G2 phase, proteins are synthesized in preparation of mitosis while cell growth continues. How do plants and animals continue to grow all their lives? Do all cells in plant divide all time? Do you think all cells continue to divide in all plant and animals? Can you tell the name and the location of tissue having cells that divide all their life in higher plants? Some cells in the adult animals do not appear to exhibit division, for example, heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury or cell death. These cells do not divide further exit G1 phase to enter an inactive stage called quiescent stage, G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so depending on the requirement of the organism. In animals, mitotic cell division is only seen in the diploid somatic cells. However, there are few exceptions to this where haploid cells divide by mitosis. For example, male honeybees. Against this, the plants can show mitotic division in both haploid and diploid cells. From your recollection of examples for alternation of generation in plants, chapter 3, identify plant species and stage at which mitosis is seen in haploid cells. Aphase. This is the most dramatic period of the cell cycle involving a major recognition of virtually all components of the cell. Since the number of chromosomes in the parent and progeny cells in the same, it is also called as equational division. Thou for convenience, mitosis has been divided into 4 stages of nuclear division karyokinesis. It is very essential to understand that cell division is a progressive process and very clear-cut lines cannot be drawn between various stages. Karyokinesis involves following 4 stages: prophase, metaphase, anaphase, telophase. Number first, prophase. Prophase is the first stage of karyokinesis of mitosis. Follow the S and G2 phase of interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centromere, which had undergone duplication during S phase of interphase, now begin to move towards opposite poles of the cells. The completion of prophase can thus be marked by the following characteristic events. Chromosomal material condensed to form compact mitotic chromosomes. Chromosomes are seen to be composed of 2 chromatids attached together at the centromere. Centromere, which had undergone duplication during interphase, begins to move towards opposite poles of the cell. Each centrosome radiates out microtubule called esters. The two esters together with a spindle fiber form mitotic apparatus. Cells at the end of prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleus, and the nuclear envelope. Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis. Hence, the chromosomes are split through the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. This then is the stage at which morphology of chromosomes is most easily studied. At this stage, metaphase, chromosome is made up of 2 sister chromatids which are held together by the centromere. Figure 2.2b. Small disc-shaped structures at the surface of the centromere are called kinetochores. These structures serve as the sites of attachment of a spindle fiber formed by the spindle fiber to the chromosomes that are moved into position at the center of the cells. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to a spindle fiber from one pole and its sister chromatid connected by its kinetochore to a spindle fiber from the opposite pole. Figure 10.2b. The plane of arrangement of the chromosomes at metaphase is referred to as the metaphase plate. The key feature of metaphase are a spindle fiber attached to kinetochore of chromosomes. Chromosomes are moved to a spindle equator and get arranged along metaphase plate through a spindle fibers to both poles. Late prophase, transition to metaphase, metaphase. Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as daughter chromosomes of the future daughter nuclei, begin their migration towards the two opposite poles. As each chromosome moves away from the equatorial plate, the centromere of each chromosome remains directed towards the poles and hence at the leading edge, with the arms of the chromosome trailing behind. Thus, anaphase stage is characterized by the following key events: centromere split and chromatid separate, chromatid move to opposite pole. Telophase. At the beginning of the final stage of karyokinesis. For example, telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosome can no longer be seen, and each set of chromatin material tends to collect at each of the two poles. Figure 10.2d. This is the stage which show the following key events. Chromosome clusters at opposite spindle poles and their identity is lost as the discrete elements. Nuclear envelope develop around the chromosome clusters at each pole, forming two daughter nuclei. Nucleus, Golgi complex, and endoplasmic reticulum differ. Anaphase. Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosome into daughter nuclei karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm called cytokinesis, at the end of which cell division gets completed. Figure 10.2e. In an animal cells, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing by cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell walls. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts in the center of the cell and grow outward to meet the existing lateral walls. The formation of new cell wall begins with the formation of a simple precursor called the cell plate that represents the middle lamella between the cell wall of two adjacent cells. At the time of cytoplasmic division, organelles, two daughter cells, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis. As a result of within multinucleated condition arises, leading to the formation of synchrome. For example, liquid endosperm in coconut. Significance of mitosis. Mitosis, or the equational division, is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells are also divided by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insects? What? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organisms is due to mitosis. Cell growth results in distributing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleo-cytoplasmic ratio. A very significant contribution of mitosis of cell repair. The cell of the upper layer of the epidermis, cells of the lining of the gut, and blood cells are being constantly replaced. Mitotic division in the meristematic tissue, the apical and the lateral cambium, result in a continuous growth of plants throughout their life. Meiosis. The production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from special diploid cells. This specialized kind of cells division that reduce the chromosome number by half results in the production of haploid daughter cells. This kind of uh, division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organism, where fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key features of meiosis are as follows. Meiosis involves two sequential cycles of nuclear and cell division called meiosis first and meiosis second, but only a single cycle of DNA replication. Meiosis first is initiated after the parental chromosome have replicated to produce identical sister chromatids at the S phase. Meiosis involves pairing of homologous chromosome and recombination between non-sister chromatids of homologous chromosomes. Four haploid cells are formed at the end of meiosis II. Meiotic events can be grouped under the following phase: meiosis first, meiosis second. Meiosis first, prophase first, metaphase first, anaphase first, telophase first. In meiosis second, prophase second, metaphase second, anaphase second, telophase second. Meiosis first. Prophase first. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior. For example, leptotin, zygotine, pachytine, diplotin, and dikinesis. During leptotin stage, the chromosome become gradually visible under the light microscope. The compaction of chromosome continues throughout leptotin. This is followed by the second stage of prophase first called zygotine. During this stage, chromosomes start pairing together, and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron microscope of this stage indicate the chromosome synapsis is accompanied by the formation of complex structure called synaptominal complex. The complex formed by a pair of synapsed homologous chromosome is called a bivalent or a tetrad. However, these are more clearly visible at the next stage. The first of two stages of prophase first are relatively short-lived compared to the next stage, that is pachytine. During this stage, the four chromatids of each bivalent chromosomes become distinct and clearly appears as tetrads. This stage is characterized by the appearance of recombination nodules, the sites at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved in called recombinase. Crossing over leads to recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of pachytine, leaving the chromosomes linked at the site of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptominal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other, except at the site of crossover. These X-shaped structures are called chiasmata. In oocytes of some vertebrate, diplotin can last for months or years. The final stage of meiotic prophase first is dikinesis. This is marked by terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of dikinesis, the nucleus disappears and the nuclear envelope also breaks down. Dikinesis represents transition to metaphase. Metaphase first. The bivalent chromosome begin on the equatorial plate, figure 10.3. The microtubule from the opposite poles of the spindle attached to the kinetochore of homologous chromosome. Stage of meiosis first. Prophase first, metaphase first, anaphase first, telophase first. Anaphase first. The homologous chromosomes separate while sister chromatids remain associated at their centromere. Telophase first. The nuclear membrane and nucleus appear cytokinesis followed, and this is called as dyad of cells. Figure 10.3. Although in many cases of chromosome to undergo some dispersion, they do not reach the extremely extended stage of the interphase nucleus. The stage between the two meiosis division is called interkinesis and is generally short-lived. There is no replication of DNA during interkinesis. Interkinesis followed by prophase second, a much simpler prophase than prophase first. Meiosis second. Prophase second. Meiosis second is initiated immediately after cytokinesis, usually before the chromosome have fully elongated. In contrast to meiosis first, meiosis second resembles a normal mitosis. The nuclear membrane disappears by the end of prophase second. The chromosomes again become compact. Metaphase second. At this stage, the chromosome elision at the equator and the microtubule from opposite poles of the spindle get attached to the kinetochores of sister chromatids. Anaphase second. It begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move towards opposite poles of the cell, shortening of microtubules attached to kinetochores. Figure stage of meiosis 2. Telophase 2. Meiosis ends with telophase second, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis flow resulting in the formation of tetrad of cells, for example, 4 haploid daughter cells. Significance of meiosis. Meiosis is the mechanism by which conservation of a specific chromosome number of each species is achieved across generation in sexually reproducing organisms. Even though the process per se paradoxically results in reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from the one generation to the next. Variations are very important for the process of evolution.